During this recorded lecture, we are going to be covering Chapter 2 in your Human Body in Health and Disease textbook. This recorded lecture will cover the content of Chapter 2, Chemistry of Life. I know that you're probably wondering what chemistry has to do with being a surgical technologist. Actually, chemistry is a very important component of understanding how the body responds to specific situations. And because we are the extra eyes and ears in the operating room, it's important that we have those basic understandings as well. Now, we're not studying to be chemists by any means, so I don't expect you to know everything that is outlined in the chapter. I will try to highlight the areas where you should have an understanding. In Chapter 1, we talked about the differing levels of organization, and we said that the first level of organization is the chemical level. So today we're going to talk specifically about the atom, which is the smallest unit of matter of anything. Now, there are some specific things that you need to understand about an atom, and the first thing is they contain three different types of particles, and these particles all have different charges. Now, in the center of the atom is what we call the nucleus, and in the nucleus there are two of these particles. The first particle is the proton. Now, the proton is a positively charged particle that is in the nucleus, and also living in there is the neutron. Now, the neutron is an uncharged or neutral particle that is in the nucleus. If we want to talk about the atom in regards to its mass or atomic mass, how much it weighs, we would add the number of protons and the number of neutrons to get the atomic mass. Now, we don't add in the electrons because those particles are so small that they really don't even contribute to the weight of the atom. Now, the other number that you need to know about is the number, the atomic number. The atomic number is the number of protons. Now, a lot of times the nucleus, inside the nucleus, the neutrons and protons equal each other, and the electrons also equal the protons. However, as compounds are made and atoms join together with each other, these particle numbers can change. It's also equally important to talk about the levels or orbitals that exist outside of the nucleus. This is where the electrons live. The electrons are arranged in shells or orbitals that circle the nucleus. The orbitals of the atom are arranged into energy levels. So the energy level is the region surrounding the nucleus that contains the electrons. The first energy level has one orbital. Now, each orbital can hold 2 electrons. Each additional energy level has up to 4 orbitals and can hold 8 electrons, 2 electrons per orbital. So if you look at this sodium atom here, you can see that the first energy level, or the circle closest to the nucleus, has 2 electrons. The second energy level contains 8 electrons because it can have up to 4 orbitals, 2 electrons per orbital. And the third energy level has that last electron. Now, sodium, if we count these up, we will find out that there are 11 electrons in sodium. Sodium also has 11 protons and 12 neutrons. So how would we determine sodium's atomic mass? Well, we would simply add 11 protons plus the 12 neutrons to give us an atomic mass of 23. What if we wanted to find out the atomic number? Remember, the atomic number is simply the number of protons. So the atomic number for sodium would be 11. Now, it's important to understand that the energy levels that are closest to the nucleus are going to have a stronger pull on them because remember, positive charges and negative charges attract. It's like having two magnets, a negative pole and a positive pole. The closer you bring the magnets together, the more you're going to feel them pull on each other. So the atom behaves in the same way, and this first energy level, the electrons are held more tightly to the nucleus, so their energy levels are going to be lower. However, as the orbitals that are further out from the nucleus, those electrons in those outer energy levels are going to be moving a lot more quickly and have a lot higher energy because there's not as much tug that they're feeling from the positive charge of the protons in the nucleus. Make sure that you're familiar with the terms element, molecule, and compound and that you can distinguish between the three. Remember, an element is a pure substance, like gold, anything that could be found on the periodic table. These substances are made up of only one type of atom. Now, when we talk about a molecule in itself is a group of atoms that are bound together. The difference between a molecule and a compound is a compound is a substance that's molecules have more than one kind of atom. So a molecule in and of itself, same type of atom bound together, compound, several molecules with different types of atoms bound together. So here's a question for you. What are the four major elements that make up 96% of the body? Do you know? They are oxygen, carbon, hydrogen, and nitrogen. Which one of these pictured here is an element? Which one is a molecule? And which one is a compound? That's right. The helium atoms on the far left represent an element, a single pure type of substance. Helium can also be found on the periodic table. Oxygen molecules, like you see here, are the same types of atoms that are bound together in a group. These happen to be in pairs. Lastly, this is a water molecule. A water molecule is considered a compound because it is a grouping of atoms of different types. Two hydrogen atoms bonding with one oxygen atom, which gives us the formula H2O. Now, we use symbols to represent different types of atoms. So here, the H symbolizes hydrogen and the O symbolizes oxygen. When we write it together to symbolize water as H2O, we call that a formula. So what exactly is an isotope? An isotope is an atom that can have different numbers of neutrons. So if you look at the picture here, you can see that the normal hydrogen atom is on the far left. It has one proton, no neutrons, and one electron. Now, the one in the middle is the first of two types of isotopes that hydrogen can form, and you'll see that it has one neutron. The second isotope that hydrogen can form has two neutrons. So, isotopes are atoms of the same element with unequal protons to neutrons. An isotope that emits radiation is called a radioactive isotope. Now, how might we use radioactive isotopes in surgery? Well, there's a couple different ways. We can use radioactive isotopes to pinpoint the sentinel node in a breast biopsy. We can also use radioactive seeds to treat cancerous tissue of the prostate, the breast, and the cervix. An important thing to remember is that atoms want to be stable. And in order to be stable, that means that they have to have a full outermost energy level, or what we would call a valence shell. So they want to be stable. That means they need a full valence shell, or they want a full valence shell. And so this is why atoms bond to one another, so that they can become more stable. Now, there's two ways that chemical bonding occurs. The first one that we're going to talk about is ionic bond, and the second type is covalent bonding. The most important thing to remember about ionic bonds and covalent bonds is ionic bonds form by one atom stealing another atom's electrons. Covalent bonds form when two atoms share their electrons. A good example of ionic bonding is seen with table salt. So table salt is formed by sodium and chlorine. Sodium, we already looked at before, has one electron in its valence shell, and chlorine has 7 electrons in its valence shell. So we talked about how an atom wants to have a full valence shell, and the closer it is to having a valence shell, the more it wants to fill that shell. So in this case of sodium and chlorine, chlorine is going to steal that one electron from sodium. And when that happens, an ion is created. Sodium loses an electron, and so it takes on a positive charge. Chlorine gains an electron, so it takes on a negative charge. We already said that negative and positives attract, so this stealing of electrons, this change of charges of sodium and chlorine, makes them attract together to form table salt. Now, a covalent bond shares its electrons. These bonds are held much more tightly together, and so they're much more difficult to break, whereas ionic bonds are much more easily broken apart in water. It's important to remember that positive ions are formed by losing electrons, and negative ions are formed by gaining electrons. Positive ions have a plus symbol that follows the atomic symbol, and negative ions have a negative sign that follows the atomic symbol. So sodium would be Na+, chlorine would be Cl-. It's also important to remember that these types of bonds break apart easily in water because water carries these negative and positively charged poles. More positively charged where the hydrogen lives and attaches, and more negatively charged up kind of on the top of the oxygen molecule. So if we were to take a teaspoonful of table salt and put it in some water and stir it around, eventually those crystals are going to dissolve. This is called dissociation. So we say that ionic bonds dissociate in water. And dissociation basically means that they break apart or dissolve easily in water. Now, because ionic bonds dissolve easily in water, they separate into their positively charged particles and their negatively charged particles. And when this happens, we refer to those as electrolytes. You might have heard of some electrolytes like potassium, magnesium, and calcium. All of these are really important electrolytes for the functioning of our nerves and our muscles. As I mentioned previously, electrolytes are molecules that dissociate or break apart in water to form individual ions. An example that I gave was of a strong electrolyte, table salt, or sodium chloride. Sodium chloride completely breaks apart into sodium and chlorine in water. Now, there are also weak electrolytes that partially dissociate and non-electrolytes that do not dissociate at all. I want to let you know that you do not need to memorize any formulas or any reactions of any type. I would just like you to have an understanding of what an electrolyte is and that there are various forms of them, and that strong electrolytes fully dissociate while other electrolytes either partially dissociate or do not dissociate at all. When atoms form covalent bonds, they're sharing their outer electrons with each other. This makes them more difficult to break apart. So typically, covalent bonds do not dissociate in water. Covalent bonds usually contain atoms such as carbon, nitrogen, oxygen, and hydrogen. They make up organic compounds in our bodies like carbohydrates, fats, and proteins. Have you ever jumped off the high dive and ended up doing a belly flop? Man, does that sting. Well, you can thank hydrogen bonds for that sting. While hydrogen bonds do not create new molecules, they do have a slightly positive charge that helps keep neighboring molecules together. So when I think of hydrogen bonding, I like to think of that as glue. So hydrogen bond is the glue that helps hold other molecules together. It's also the glue that helps water molecules pack tightly together and deliver that sting when you hit the water. So hydrogen bonding can be found in many different situations in the body. One example is with proteins. So when proteins are made and hydrogen is part of that protein as it is assembled, those areas where there is hydrogen cause the protein to fold, like you see in this top picture here, these little curly cues. That is because of hydrogen. It's also what gives DNA its helix type properties, is because of these hydrogen bonds. So all compounds can be classified in two ways. One is as organic compounds and the other is as an inorganic compound. So organic molecules typically contain carbon and hydrogen. Now, whether the carbon is bonded to another carbon or the carbon is bonded to a hydrogen, this bonding is a covalent bonding. So this is a very strong bond that is created between carbons and hydrogens, or carbons and carbons, which makes them very difficult to dissociate or dissolve in water. Some examples: table sugar, DNA, methane, ethanol. Other examples: carbs, fats, and proteins. The first inorganic compound we're going to talk about is water. Now, water is one of the most common solvents that we use to dissolve substances in. So the liquid component, water in this case, would be considered a solvent, and any type of compound that we were going to be dissolving into the water would be considered the solute. So the solute could be salt, and the solvent could be water. And when we mix them together, the water, as we said before, dissociates, the salt dissociates in the water, and we are left with some sort of solution. An aqueous solution that we already talked about is 0.9% sodium chloride, or saline. Saline is 0.9% saline is the closest composition to the fluids that are inside and outside of our cells, which makes it safe for us to irrigate wounds with. So I know you're all wondering, what is Mrs. Windsor's favorite solvent and solute? And the survey says coffee. I like a little cream in there too. There are two common types of chemical reactions in the body that have to do with water. The first one that we're going to talk about is dehydration synthesis. Have you ever heard of a dehydrator? If you have, then you'll understand that when you put fruit or vegetable or meat into a dehydrator, water is being removed from that to dry it out. This is similar with dehydration synthesis. Water is removed when two molecules are combined. Now, it's important to remember that dehydration synthesis builds bigger molecules by removing water. An example of this is when two monosaccharides join together to form a disaccharide. So a monosaccharide is a single unit of sugar, and a disaccharide is 2 units of sugar. So when two monosaccharides join, a water molecule is formed when those two molecules can join together and form a disaccharide. Water is removed so the two monosaccharides can join to form a disaccharide. We're building a bigger molecule by removing water. The second type of reaction is called hydrolysis. Remember, for medical terminology, hydro means water, lysis means the condition of breaking something down. So we're using water to break something down. This is opposite of dehydration synthesis. Remember, dehydration synthesis builds bigger molecules by removing water. It allows them to hook together. Hydrolysis uses water to break bigger molecules apart. A good example of this is how the body breaks down suture material that we use to close wounds or suture tissues together. The body will utilize water to break apart the material that the suture is made of. So again, dehydration synthesis removes water so that smaller molecules can hook together to make bigger ones, and hydrolysis uses water to break bigger molecules into smaller parts. As discussed on the previous slide, hydrolysis is the process that the body uses to break down different types of sutures in the body. Now, not all sutures are broken down readily in the body. There are two different types of suture materials. There are ones that we consider absorbable that break down in a reasonably short amount of time, whether it's a few days or a few months. And then there are non-absorbable materials such as silk, or surgical steel, or cotton, or nylon that take a very long time to break down, or maybe the body might not ever break them down. That doesn't mean that the body doesn't continue to try. However, this is just another example of how hydrolysis can be used in the body. Now we're going to talk a little bit about acids, bases, and salts. Now the book goes into quite a bit of detail regarding reactions and formulas. You don't need to know those. The important takeaways for us are just to know the basic differences between them. So the first difference that I want to point out between acids and bases is the level of hydrogen ions to hydroxide ions. Now, hydrogen ions we already know is represented by an H with a plus. Hydroxide ions are represented by an OH negative. So OH negative is hydroxide and H positive is hydrogen. Now, as chemical reactions occur in water, they produce varying levels of either hydroxide ions or hydrogen ions. When we have a situation where a solution is extremely acidic, that means that it has a high level of hydrogen ions. It also means that it has a low level of hydroxide ions. The opposite is true with a base. With a base, there is a higher level of hydroxide ions and a lower level of hydrogen ions. Here we see the pH scale ranging from 0 being the most acidic to 14 being the most alkaline or basic. Anybody know what the P stands for in pH? It actually stands for potential. So here we have a scale of potential hydrogen. As we move up or down the scale, we're increasing or decreasing levels of hydrogen ion concentration by factors of 10. So if we look at a pH of 6 and we're moving to a pH of 5, we're increasing the level of hydrogen ions by 10. Now, what if we move from a pH level of 6 to a pH level of 3? How many times are we increasing the hydrogen ions? If you said by a factor of 1,000, you would be correct. Now, if we look at this pH scale, 7 is going to be our neutral zone. So 7 is neither acidic nor basic or alkaline. But as we move down the scale towards 0, our hydrogen ions increase by factors of 10. As we move up the scale from 7, our hydrogen ions continue to decrease by factors of 10. When we're talking about normal pH levels for blood, blood is slightly alkaline with a pH of 7.35 to 7.45. Now just a quick shout out to salts. The important takeaway here and what I'd like you to know about salts is when a strong acid and a strong base is mixed together, there are typically two products. The products are water and salt. Like I mentioned before, the normal pH of blood is 7.35 to 7.45. Now, the body typically does a really good job of maintaining this normal pH level. However, there are critical situations that the body can be put in. One is called acidosis, which is the condition of low blood pH, and alkalosis, which is the condition of high blood pH. One of the ways that the body addresses a pH that is too acidic is by excreting hydrogen ions in the urine. Another thing that the body can do is increase respirations to expel carbon dioxide. So let me give you an example. An example is you decide to do 50 jumping jacks. After you have been doing jumping jacks for a while, your respirations are going to increase. Your breathing is going to get faster. This is because when your cells burn energy, or ATP, a waste product of that is carbon dioxide. Now, when carbon dioxide mixes with water, it forms carbonic acid. Now, carbonic acid increases the pH level, or decreases the pH level of the blood, making it more acidic. So one of the ways our body compensates is by increasing respirations, which does two things. One, it provides oxygen so that the body can continue to create energy for its cells. And two, it allows for the expiration of carbon dioxide, which helps to bring that pH, that acidic pH, to a more normal body pH. Another way that the body can maintain homeostasis is through the use of buffers. I don't need you to know too much about buffers, just that they are one way that the body can maintain homeostasis of pH. Now we're going to switch gears and talk about organic chemistry. Organic chemistry includes compounds like carbohydrates, lipids, which are fats, proteins, and nucleic acids. Nucleic acids make up DNA and RNA. Now, when we're talking about carbohydrates, we're basically talking about sugars in different forms of complexity. So, carbohydrates are our body's favorite type of substance because it's pretty easy for the body to break down carbohydrates into simple sugars that can be utilized for energy in the cells, or either stored and used for energy later. Organic compounds, like we said before, contain carbon-to-carbon bonds, carbon-to-hydrogen bonds, and a lot of times they contain oxygen as well. Now, the most simplest sugar is referred to as a monosaccharide, and it's the source of energy that's used by our cells. Examples would be glucose, galactose, and fructose. Disaccharides, again, I mentioned earlier, are two monosaccharide units that have been joined together by, do you remember what process? Dehydration synthesis. Right, very good. So disaccharides are maltose, sucrose, and lactose. So milk sugar, table sugar. Lastly are polysaccharides, which are the most complex carbohydrates that are made up of several chains or several units of monosaccharides joined together. And some examples of these are glycogen and starches like pasta, bread, and potatoes. This slide gives you a visual of what the various types of carbohydrates look like. The first one being that single unit sugar, monosaccharide, and then two monosaccharides joined together would be a disaccharide, and more than two monosaccharides joined together would be a polysaccharide chain. Fats are also referred to as lipids, and there's three different types of lipids commonly found in the body. There are triglycerides, phospholipids, and steroids. Now, triglycerides have a glycerol head and three fatty acid tails. There are two types of triglycerides, saturated and unsaturated. So you may have heard different products referred to as having saturated fats or unsaturated fats. Now, unsaturated fats are the fats that are more healthy for us, and some examples would include olive oil or something like that. Now, when we're talking about saturated fats, those are fats that solidify or are solid at room temperature. So unsaturated fats are not solid at room temperature. Saturated fats are solid at room temperature. The difference between the two is how the molecules hook together when they're forming their fatty acid tails. So with saturated fats, you can see this first example of the triglyceride where the tails are very straight. That enables them to pack together very closely, whereas the unsaturated fats have these fatty acid tails that are kind of crooked, so they can't pack together as closely. All right. The body can also use lipids as a form of energy, or triglycerides rather, as a form of energy. However, it's their second favorite form of energy next to carbohydrates. Phospholipids are a type of lipid that are composed of a glycerol, a phosphate, and two fatty acid chains, like you see in this top picture here. The really cool thing about phospholipids is they make up something called the phospholipid bilayer, which is the main structural component of our cells' membranes. What's really cool about these guys is that their glycerol heads are hydrophilic. So that means water is attracted to them and they're attracted to water. Now their tails, on the other hand, their fatty acid tails, don't like water, and they repel water, so they end up in this formation where the heads are facing out and the tails are facing in. So if you think about a cell, outside the cell is surrounded by water, and inside the cell is surrounded by water. So these phospholipids arrange themselves where their heads are positioned towards the inside of the cell and towards the outside of the cell, making this double layer or bilayer that we call it, and their tails are on the inside. Another type of lipid is a steroid, and a good example of a steroid is cholesterol. Now here in this image you see that phospholipid bilayer, and these cholesterols position themselves within the phospholipid bilayer. They kind of wedge themselves in there. Cholesterols perform several critical functions of the body, and one of the things that they do is to help stabilize the structure of the phospholipid bilayer. So that's really awesome. They also are these multiple ring structures that make up the building blocks of hormones like cortisone and estrogen and testosterone. Another lipoprotein that you probably hear a lot if you go to get your blood tested or get a physical of some sort, they might talk about low-density proteins, low-density lipoproteins, and high-density lipoproteins. Low-density lipoproteins are bad. They're bad because they actually carry cholesterol into the cells, and that cholesterol gets carried into the bloodstream and it collects on the walls of the vessels. This can lead to something called atherosclerosis, or fatty deposits in the walls of the arteries that cause hardening of the arteries. High-density lipoproteins are good because they carry cholesterol away from the cells towards the liver so that it can get eliminated from the body. Okay, let's talk about proteins. Proteins are large molecules that are made up of these little building blocks called amino acids or peptides. Now, these amino acids or peptides, they join together by something called peptide bonds, and they create peptide chains. Now, there's different structures and levels of these proteins. You don't really need to know about primary, secondary, tertiary, quaternary. However, it is important for you to know that their shape determines their function. So that's a very important thing to consider when you're thinking about proteins. So some different types of proteins, 3 different types we're going to talk about. The first type is structural proteins. So structural proteins form essential structures of the body. Some examples are collagen and keratin. So collagen holds majority of the body tissues together, while keratin forms waterproof networks in a number of the layers of the skin, the nails, the hair. Now, functional proteins participate in chemical processes of the body. These are different types of hormones, growth factors, cellular membrane channels that move different types of atoms and components in and out of the cell. And then lastly, enzymes. So enzymes cause something to happen. So we refer to them as a chemical catalyst. They participate in chemical reactions. They're not changed by the reaction, but they change a molecule or a substrate. So they will join to a substrate, and they will, which is a substance that an enzyme acts upon, and they will change it somehow. This is considered a lock and key model where only a specific enzyme, aka key, will fit onto a simple substrate, aka a lock, a key and lock model. Lastly, coming into the home stretch here, we're going to talk about nucleic acids. So there are 2 forms of nucleic acids. One form is deoxyribonucleic acid, DNA, and the other type is ribonucleic acid, RNA. Now, DNA and RNA are the genetic materials that are located in the nucleus of every single cell, and they're basically the instructions to make and replicate everything within that cell. So it's pretty important recipe book for our cells and our bodies. Now, each nucleic acid, DNA or RNA, is made up of these smaller components called nucleotides. Nucleotides are composed of a phosphate unit, a sugar, and a nitrogenous base. Well, there are 5 types of nitrogenous bases that we're going to talk about, but first let me circle back to the sugar. When we're talking about sugars, RNA has a ribose. That's why it's named ribonucleic acid. That's how I remember. And DNA, deoxyribonucleic acid, has the deoxyribose sugar. So those 2 should be pretty easy for you to remember. Now let's talk about the bases. We're talking about bases. Something occurs with RNA and DNA called complementary base pairing. So you'll see this image here on the slide where there's different colors of bases: orange, blue, red, green, yellow. And you can see a strand of RNA and a strand of DNA. Now, the names of the nitrogenous bases are adenine, thymine, uracil, guanine, and cytosine. Now, specific bases pair with a specific partner. So when we're talking about DNA, DNA has adenine, and that pairs with thymine, guanine, and that pairs with cytosine. Now you'll see that the guanine and cytosine complementary base pairing is the same for RNA. However, there is one difference with the adenine. Now in RNA, thymine is replaced by uracil. So adenine bonds with uracil or pairs with uracil in the RNA nucleic acid. So have an awareness of what these two different types of nucleic acids are, that they do have these complementary base pairings, what they are and what they pair with. For our participation activity, we're going to focus on the P53 protein, so stay tuned for that. We saved the best for the very last, and that is adenosine triphosphate. Now, adenosine triphosphate is a molecule that allows cells to make energy. All right, so you can see in this diagram here that there is one adenosine and 3 phosphates in one group. So there will be more to come on this in later lectures. However, just know for now that adenosine triphosphate is responsible for energy transfer in our cells, and the way that that happens is a phosphate will break off from the adenosine triphosphate, and that yields so much energy, and then that becomes adenosine diphosphate, and that regenerates to a triphosphate, and the energy happens over and over again. So ATP can also be seen as a type of battery that stores energy for our cells. So you made it. That concludes our lecture on the chemistry of life. Make sure you identify any muddy point questions that you have, and I'm looking forward to seeing you in our Zoom session.